अब हम बात करेंगे सेकंड मेथड की, जो कि यूज़ किया जा सकता है प्योरिफिकेशन और वॉशिंग ऑफ इंक्लूजन बॉडीज़ की। इसमें वी आर गोइंग टू यूज़ यूरिया एज अ वॉशिंग reagent instead of Triton X100। स्टेप नंबर 6 से हमें जो सेल लाइजेट अवेलेबल हुआ था उसी को लेके हम आगे चलेंगे, और उस सेल लाइजेट को जब हम सेंट्रीफ्यूज करेंगे स्टेप नंबर 7 में, वॉशिंग के पहले स्टेप में, देन वी आर गोइंग टू बेसिकली सेंट्रीफ्यूज इट एट मैक्सिमम स्पीड फॉर 15 मिनट्स एट 4 डिग्री सेंटीग्रेड। जैसा कि इस सेंट्रीफिकेशन स्टेप के हवाले से मैंने पहले इसका पर्पस बताया कि हमें इसमें से अनवांटेड सेलुलर डेब्रीज और अनवांटेड मटेरियल्स को रिमूव करना है, इसलिए हमें ये स्टेप परफॉर्म करना पड़ रहा है ताकि हमारे पास सिर्फ प्रोटीन्स जो हैं वो पीछे बाकी रह जाएं लाइजेट में। वंस दिस सेंट्रीफिकेशन स्टेप इज़ ओवर आफ्टर 15 मिनट्स, we are going to decant the supernatant and we are going to proceed further with the pellet. The pellet is going to be resuspended in 1 milliliter of water. 1 milliliter of water is going to be added and pellet is going to be resuspended using the micropipette or using the glass rod mixing or any other method. Now this resuspended pellet is going to be processed further once we determine which concentration of urea is going to give best washing. For that purpose, we are going to take 100 microliter from this resuspended pellet in 4 different new tubes, the microfuge tubes. So 100 microliter from the resuspended pellet in 4 new microfuge tubes is going to be shifted, and the rest of the volume or the rest of the resuspended pellet will be stored at 4 degree centigrade. Again, centrifugation step so that we can separate the proteins. 100 माइक्रोलीटर लिक्विड जो हमने 4 डिफरेंट ट्यूब्स में लिया है उसको हम सेंट्रीफ्यूज करेंगे 15 मिनट के लिए 4 डिग्री सेंटीग्रेड पे, ताकि लिक्विड पार्ट और वाटर जो है उसको हम रिमूव कर सकें। सुपरनेटेंट इज गोइंग टू बी डिस्कार्डेड। यहाँ पे वी आर गोइंग टू डिस्कार्ड द सुपरनेटेंट because that is water, we don't need it anymore, and we are going to resuspend the pellet in 100 microliter of 0.1 molar tris Cl, pH 8.5। अब ये tris Cl जो है that is variable because of differences in the urea concentration that starts from 0.5 molar and goes up to 5 molar. So we are going to add different concentrations of urea in each tube, and then we are going to proceed further. Again, another centrifugation step so that we can separate the tris Cl and we can proceed further towards the extraction or purification step. Centrifugation for 15 minutes at 4 degree centigrade will be performed at maximum speed. The same protocol, the same centrifuge machine is going to be used. Now decant the supernatant. Now this supernatant is going to be important. We are going to decant, or we are going to transfer this supernatant in a separate tube, and we are going to resuspend the pellet again in 100 microliter of water. Now at this stage again we have 2 samples from each dilution or from each concentration of urea. स्टार्ट पे हमारे पास 4 माइक्रोफ्यूज ट्यूब्स थी, अब हमारे पास 8 माइक्रोफ्यूज ट्यूब्स हैं, एंड ईच वन is having the supernatant, the second one is having the resuspended pellet, and these two, the first two step is from the 0.5 molar concentration of urea, 1 molar concentration of urea, 2 molar and 5 molar. From each of this tube, we are going to take 10 microliter of sample. This 10 microliter of sample is going to be mixed up with the 2x sample loading buffer or gel loading buffer. And we are going to test, or we are going to check which supernatant or which resuspended pellet gives us the best result regarding our protein of interest. For that purpose, we are going to perform SDS page. 10% SDS page is going to be performed, and the results are going to tell us that which concentration of urea is best for washing, and we should proceed further for that particular concentration using the lysate, which at the start of this step we placed at 4 degree centigrade. अब उस लाइजेट को लेके जो यूरिया की कंसंट्रेशन हमने डिटरमिन की है, हम इस वॉशिंग प्रोसेस को रिपीट करेंगे ताकि हम मैक्सिमम प्रोटीन, फॉरेन प्रोटीन को आइसोलेट कर सकें. यहाँ पे अगेन द सेम थिंग, इफ़ वी आर required to perform solubilization of inclusion bodies, then we may need to perform some additional step. Otherwise, up till this point, we are successful in separating and purifying our foreign protein of interest, which was expressed in a bacterial cell as a result of transformation using a recombinant plasmid that carried the gene of interest which we inserted in it.